All right, let's get started. Excited for another edition of CRISPR Office Hours. First off, welcome to CRISPR Office Hours. Thanks for joining us today. We're really excited as we're starting season 2, episode 1. And if you want a recap of season 1, you can just go to our blog, synthego.com/blog, and you'll get to see the first season recap with the first 5 episodes and all our guests that we've had on. So we're really excited to host CRISPR Office Hours as we've been doing it on a weekly basis. This is episode 6 or season 2, episode 1. And we host CRISPR Office Hours weekly to help the genome engineering community navigate these unique and challenging times. So just for some housekeeping items, we want to make sure to let everyone know that they're on mute by default, and we prefer you stay that way. If you have any questions, please use chat to send in your questions. We want to make the session as interactive as possible, so the more questions you can send in, the more interaction we have, the more value that everyone on the office hours gets from each other. And the other thing is this will be recorded and uploaded to our YouTube channel. We have the past 5 office hours on there as well. So as we're going through CRISPR Office Hours, we always want to make sure that there's a chance for the community to hear from one another, let the genome engineers, research scientists, other life science professionals discuss and better understand how the COVID-19 pandemic is impacting their research. So as we said earlier, the more interaction, the more questions you have via chat, the more value we all get from it together. So with that to start off, my name is Aditya Vempati, and I'm the VP of Marketing at Synthego. And as always, my talented co-host who just celebrated a birthday yesterday, Kevin Holden, is on with me.
Hey, Aditya. Yeah, thanks for coming on, everybody, again, and welcome to another exciting episode of CRISPR Office Hours. So, yeah, I'm your co-host, Kevin Holden, Head of Science at Synthego. The title for our CRISPR Office Hours episode this week is Keep Calm and CRISPR On During COVID-19. Researchers are raring to go. All right, so as we've mentioned a few times in the past few weeks, now has never been a better time to keep calm and carry on. So this was originally a poster that was made by the British government during World War II as a part of a public assurance campaign to remind people that even in the face of incredible danger, it's important not to be paralyzed by fear, but to keep calm and get on with what you need to do. So we think this slogan has never been more appropriate than these times we're living in now, as we all do our part to try and fight the COVID-19 pandemic. And, we just like to remind you all that from Synthego, keep calm and CRISPR on, and think about how you're going to reignite your research using CRISPR once you come back into the lab. All right, so Aditya, you want to tell our friends in the audience how they can get one of these cool t-shirts?
Yeah, so we want to say a special thank you to everybody as we want them to keep calm and CRISPR on during these times. So at the end of the office hours, we'll let you know what URL you can go to claim your shirt. So with that being said, let's meet today's panel. So as always, my co-host, Kevin Holden, Head of Science at Synthego. You can reach him at his Twitter handle, Kevin Holden SF, but our guest and part of our panelists who I'm really excited to speak with today from Down Under, Imran House, Ph.D. He's a postdoc at Peter MacCallum Cancer Centre, and his Twitter handle is Imran House. Pretty simple. And myself, Aditya Vempati, I'm the VP of Marketing Synthego. My Twitter handle is AVEMII. Much more complicated than the rest of them, obviously, but makes for fun times. So with that being said, welcome, Imran.
Thank you. Thank you very much for having me. It's a little bit early, I have to say. It's 10 past 2 in the morning, I think, here, which is the earliest I've ever given a talk. I gave a talk for Synthego at 5 a.m. the other day, and this is a little bit more insane than that, I have to say. But, it's great. My cat is very confused.
Still late, right?
Late. I went to sleep and got up, so it's,
okay.
Well, yeah, my cat is very confused, as I said. Why am I awake at 2 a.m.? No idea. But
you're encroaching on your cat's hours.
Yeah, he's here with us.
There you go. Cool. So, Imran, we always start off with a poll, try to get the audience engaged. get them warmed up and ready to, or raring to go as the talk is. So today's poll, we're thinking about it. It's been a while. We've been in quarantine, various parts of the world. Some people are getting out. And today's poll question was, what's your preferred food delivery service? And we have a few options here. 1, Uber Eats. 2, DoorDash. 3, Deliveroo. Apparently this is a service in Australia that Imran's let us know. Grubhub. No contact delivery from grandma. 6, drive-through. 7, whatever's in my freezer. So let me ask you, Imran, what's your preferred method?
So I feel like definitely Uber Eats. I think it's just generally always Uber Eats here, but Deliveroo isn't like an exclusively Australian thing. I first heard of it in Germany, I think, but yeah, so it's not just kangaroos delivering food, although that is a fantastic idea. And if someone hasn't done that yet, probably should get on it.
What about yourself, Kevin?
I'm just gonna have to go with whatever's in the freezer. So I actually enjoy cooking a little bit. So most of the days it's just preparation, but if I can yank something out of the freezer and cook that, it's gonna be easier for me.
I mean, I'm between a combination of Uber Eats and whatever's in my freezer. Uber's definitely got me on the hook with all the credits and no delivery charge and wanting to help the local economy. So let's see what our attendees are saying. A lot of folks here are saying, I love cooking. I love fresh food, homemade always. We got a few Uber Eats credits via CSR. Okay, from Zoe. Cool. There's definitely a lot of cooking at home. That's nice to see. Not as much as Uber are taking out as I thought there would be. So that's cool. All right. With that being said, I want to say thanks. And then as we go forward here, I want to let Kevin talk about the model we've been discussing for the past few weeks, about 5 weeks or so. And I think we're entering a different phase here. I'll let Kevin share a little bit more on this.
Yeah, sure thing. So for the past few weeks, we've been discussing this model that was developed by the Linus Group, where they surveyed over 2,000 scientists about their thoughts and activities during the pandemic. So for some of us, depending on where we are in the world, it feels like we're kind of moving into this transition phase, right? Like things are starting to open up. So last week we heard from Laura Lambert at University of Alabama in Birmingham about how she's coping during this time and juggling her research, running a core facility, and taking care of her family. And so today we're bringing in Imran, a scientist from Australia in Melbourne, he's had quite an adventure to get back home right before the shutdown and has basically been raring to go ever since. So Imran's going to tell us a bit about his own research and how things are going where he is in his part of the world now that the shutdown's actually technically ended in Australia. Okay, Imran, so thanks again so much for being on the show today. Can you tell us a little bit more about yourself, who you work for, and where you work?
Yeah, no, that's great. And thank you very much for the introduction and opportunity to speak. So yeah, as stated, I'm a postdoc at the Peter MacCallum Cancer Centre here in Melbourne, which is the biggest cancer hospital in Australia. So it's half cancer hospital and half research institute. And that's really a great place to be for getting translational outcomes for patients and really, really awesome research outcomes. So I did my PhD finishing back in 2016, which seems like quite a long time ago, where I worked with Joe Trapani and Ilya Voskobojnik, and where I was focusing mostly on killer cell cytotoxicity. So Ilya's lab is actually called the killer cell biology lab, and these are cells that have the capacity to go and kill other cells, and we'll talk about them a little bit more in a moment. And as a part of that, my PhD focused on a disease or diseases that arise when patients' killer cells don't work properly, right? And this disease is called familial hemophagocytic lymphohistiocytosis. So it's really easy, just rolls off the tongue quite nicely. But it got my interest in primary immune deficiencies going. So it was then that I moved to Munich to work with Professor Christoph Klein, who is really one of the world leading researchers in primary immune deficiency work. And during this time, this is when my CRISPR work picked up a lot, because I will probably get to this, but a lot of the CRISPR applications is fantastic for looking at immune cells and looking at immune cell dysfunction. and also potentially gene therapy applications for addressing these disorders, which is now really a good part of what I do is using CRISPR for gene engineering applications. And then if we can go to the next slide, now in 2018, so this is Dr. Paul Beavis, and this is actually the Peter MacCallum Cancer Centre. This was opened by Joe Biden, actually. So he came and visited back in the day when he was vice president and cut the ribbon on the building. And so I now work for Dr. Paul Beavis. And so Paul and I were friends before I moved to Germany, and we play cricket together. So this is a photo of us playing cricket. And then he's just a fantastic researcher as well. So he got this opportunity to become a group leader, and it was a bit of a no-brainer. So I came home quicker than expected, I think, but it's fantastic to be working with Paul now. So if you go to the next slide.
Yeah, Imran, maybe you can tell us a little bit about the immuno-oncology work you do.
Yeah, so yeah, that's what we do. So our work is broadly oncoimmunology and looking at immunotherapies. So these are immune-based therapies where the immune system can be reactivated or activated in one way or another to kill off and kill cancer cells. And so people may be familiar with immune checkpoint blockade. I'm not actually going to talk about that today because we can't speak forever, but a good part of our work is looking at how therapies such as immune checkpoint blockade actually work, and the things that are essential for those therapies to work. But one of our major applications for our CRISPR technologies is in the use of chimeric antigen receptor T cells. So this is for those who aren't familiar, this is going to sound like something out of a fantasy tale, but trust me, it's a real therapy that is actually used. So this diagram, it's been a little bit squashed actually, I just realized on my slides, but so the idea is where you take from a patient, you isolate their T cells. So these are those cells I was telling you about that have this killer capacity, so the capacity to kill other cells. And you transduce them, so with a virus, you genetically modify them, so they express a chimeric antigen receptor. So that's, if you just maybe a digit point out there, this is this receptor. So it's actually a chimeric antigen receptor. And these are crazy receptors that what they have is a recognition domain, which allows the T cell to have the capacity, has the capacity to recognize an antigen expressed on a tumor cell. And then the other side of that is our signaling domains, and generally from CD28 or CD3, there's a few different signaling domains that can be used. So when the T cell recognizes that cancer cell, they activate, release cytokines, and also release killing molecules to kill off that tumor cell there. And if we go to the next slide, yeah, so these therapies have been incredibly effective at treating blood cancers. So these are some of the key publications that have looked at this work, and this is generally treating and targeting an antigen known as CD19, and it's in many cases is leading up to a 90% complete remission rate. Excuse me, as can be seen in the top left-hand corner. And importantly, this was relatively recently FDA approved, which is the next big stamp that comes along. So it's really, really quite an exciting technology, but there's a big but. If we go to the next slide, please, thank you. This is, you're not meant to be able to, yeah, that's right. If you're not meant to be able to read this table, the point is here, there's a lot of different clinical trials that have gone ahead trying to use CAR T cells in solid tumors. So these aren't cancers that are floating around in the blood, but rather this is a tumor that's a cancer that's forming a mass in the patient. And CAR T cells, we've tried for a very, very long time, but they just are nowhere near as effective in solid cancers as they are for blood cancers. And that's one of the key research focuses at the Peter Mac, but also in Paul Beavis's lab, is how can we get CAR T cells to work more effectively? And can we potentially see the same clinical benefit we see for blood cancers as we do in solid cancers? And if we go to the next slide, please, we'll see that there's a lot of reasons why they might work really well in liquid cancers. So this is a little bit simplified here, but let's just call them liquid cancers for now, liquid cancers compared to a solid cancer. So as you can see in the diagram on the right there, we have CAR T cells that are killing a liquid cancer, and they're floating around, and they have the ability to, one, traffic to the site, two, recognize their single antigen, and then eliminate that cancer. But on the left, we have a solid tumor. So immediately you have to think, well, actually this CAR T cell has to make its way to the tumor. It has to know that the tumor is there, and then make it into the actual tumor mass, which is a challenge that isn't faced when killing off a blood cancer. We also then have this concept of tumor heterogeneity. and I apologize, I just realized all my Australian spelling. That is not, they're not awful typos, it's just how we spell it, I'm sorry. Tumor, T-O, yeah, that is correct. Anyway, yeah, so tumor heterogeneity. So we send in CAR T cells and they can recognize one antigen. But what we know happens is that the CAR T cells will kill the tumor cells that express the antigen, but they won't kill, they don't have the capacity to kill the tumor cells that don't express that antigen. So what you essentially do is you just get an outgrowth of antigen negative tissue, which is a challenge that we face. And the third one, which really I think is the most well addressed with CRISPR applications, is the immunosuppressive tumor microenvironment. So a tumor is a nasty place to be if you're a T cell. A lot of the time, it can be quite, it's full of immunosuppressive signals, which really dampen their function. So I think I have a very busy diagram on the next slide. Please, thank you. And now you're not meant to, this is from Amanda Oliver, one of the PhD students in our lab, a nice review that she did. And you're not meant to understand all of this, and that's kind of the point. The whole point is that you have your effector T cell, which you probably can't even see. It's the little purple one in the middle. When it goes into the tumor, it's just bombarded with immunosuppressive signals, right? So there's TGF-beta is a big one that people are probably familiar with. You also have IL-10. You have immunosuppressive metabolites such as adenosine, and a bunch of other tumor cells. So the tumor cell makes an environment, a tumor mass makes an environment that is just formidable to be for a T cell. Here, though, is the potential application for CRISPR, and really where I apply a lot of my CRISPR work. So I just have a subheading if you just want to click to the next line. But what we can potentially do is use CRISPR technologies. That a lot of these immunosuppressive molecules are detected by receptors. So we can remove those receptors and re-enhance T cell function. Also, from a research application, and something that I do is you can knock out the suppressive molecules that are being generated by tumor cells and see the effect on T cells. So, in a recent publication, I wanted to address, for example, whether a certain chemokine was important, or tumor-derived chemokine. And it's incredible, back in the day, that would have been a really, really significant problem to do that. But it was just a matter of getting a couple of guides, knocking it out, and injecting it back into the animal. But and then looking at the immune response. So we also, though, of course, use this. It's now in vogue that people are knocking out genes in CAR T cells and adoptive cellular therapy. So other T cell therapies, knocking out receptors such as PD-1, which responds to suppressive ligands such as PD-L1. And it's really, really exciting to see where we can go with the field there. So I haven't shown any data today, it's all stylistic, but please ask me questions about any of this work and some of the more specific applications of what we do if you'd like to hear about them.
So Imran, yeah, thanks for giving us a great overview of the work you're doing in CAR T and understanding the immune system role in cancer. So I know back in February and March, you came out to California, like this sort of like a visiting research fellow. Can you tell us why you were there in California, where you went to? And then you also plan to go to New York City as well, right? Around that time.
Yeah, so it was a little bit interesting. We have really stepped up our editing applications, and so of course one of the really cool applications of CRISPR that people may be doing or familiar with is you can deliver with pretty extraordinary precision huge chunks of DNA wherever you want them to be delivered. And so a number of different research groups have worked on this for CAR T cell function. And what you could actually do is deliver the CAR itself, right? So the actual receptor itself to T cells. And one of the world leading groups in this homology-directed repair approach is the group of Matthew Porteous at Stanford. And so I arrived in late February. So February, I think, 28th, and I was there for 3 weeks working with Volker Wiebking, and he really helped me get my HDR efficiencies up a lot. So this is really exciting. So we now be able to deliver the CAR to our the actual CAR to our T cells, and then potentially other molecules that could potentially modulate their function as well. So this is in the field is called armoring. So you're armoring your CAR, and so you can add other molecules that enhance their function and also remodel the immune environment that they go into. So I was meant to, after that, I have a colleague who did his PhD at the Peter MacCallum Cancer Centre and then moved to New York. And it did coincide with my 30th birthday. So I thought at the end of my trip, I would go over there and spend a nice weekend with my two mates over there, Maddie and Simon. And unfortunately, as we all know, the situation escalated pretty quickly at that point. So that was the 16th and 17th of March. And Simon was pretty sick, I think, when he was there. I think he just had a cold, luckily, at the time. And so we ended up pulling the plug on that one. But it was incredible, you know, to be going over to the States on the 28th. And when we booked the flights and when I left, I think I was concerned, you know, mildly concerned, but then it really ended up in a bit of a panic situation and getting out. So I still went to New York because it was going to be so crazy expensive just to fly straight from California. So I you know, went with my original travel plans, which now on reflection does seem a little bit foolish, but you know, probably would have been okay to spend the $1,700 to get back. But
what was it like, your journey back into Melbourne, Australia? I mean, I think, did you say, because Australia shut the borders, right?
Yeah, so we shut the borders very shortly after that. Obviously, citizens can come back and permanent residents could have come back, but they put in very strict measures. I think, you know, everyone, what we have emotionally experienced in the last little while is an emotional exponential increase, right? So it went from being a little bit nervous to, I'm not sure if the restrictions are the same here, but anyone who came into the country from about the 16th of March or maybe 15th of March, here in Australia, had to go into mandatory quarantine. So I arrived home on about, it was on the 19th of March, and I had to go into my apartment, close the door, and could not leave. And so my apartment is relatively small. My wife left. She left and went to an Airbnb nearby. So because she was still going to work at that time. And so I was in by myself. She delivered food at the door. I didn't see her because it was just kind of easier to pretend that she didn't exist. She delivered food at the door, and that was existence for 14 days. And in that time, obviously the shutdown had continued. We were not under a mandatory shutdown for business. So the Prime Minister had a message saying, we have essential work that's going to continue. Is your job essential? If you have a job, your job is essential, was his line. So we could have kept going back with research, but obviously, we really could have, from a legal perspective, kept going. But luckily, you know, we're a cancer hospital as well. So, you know, the Peter Mac really put very, very tight restrictions on the research. So it was, everyone was doing dry lab work and doing their skills. But I went back to a situation where I couldn't go back to work at all. So yeah, it was certainly a different world. Everything changed very, very rapidly. And yeah, having left Australia in February, it's obviously not been the same. It's not been the same anywhere in the world. Yeah.
So what do you think, from your perspective, sounds like things weren't as bad there as the rest of the place? Why do you think that is?
Yeah, there's a few reasons. I think there was very early response. So we China is one of our major trading partners in the world. So and we are very reliant on for our education institutions here on students coming from all around the world, but a large part of that is coming from China. So there was very early restrictions, and I can remember, you know, not really understanding the situation at the start because there was restrictions on travel from China, and I was like, well, they've got to open that up soon because otherwise the students can't come in. And you know, that sounds obviously so crazy now because of no one being able to travel. I think we had that as an advantage, so that happened quite early. We also have an island which gave us a bit of a delay, and sadly, I guess because things progressed so rapidly in Europe and particularly in Italy, that the government was able to react. And also we are kind of, I think, dragged along by New Zealand government. I'm not sure if anyone's familiar with Jacinda Ardern from New Zealand, but her response and the country's response has been pretty incredible. So, and Australia reflected that, and the borders were shut to travel very, very early, and the people were going into mandatory quarantine. And another part of that, and I'm not sure if that's a similar situation, if you were to, I think it was after maybe, I want to say the 22nd and around that, because they realized that people weren't quarantining properly, you got taken to a hotel and you were there for 14 days. And that's still the case if you want to come back to Australia. So there's a few expats coming back. You get taken to a hotel and you're there for 14 days. You don't go home do not collect $200 and do not pass go. You're in your hotel. So I think that kind of as a measure has been really, really helpful. And obviously all the social distancing laws and a very strong message as from our state authorities to stay home. And if you can be home, be home. And people have done that and they've complied. Luckily, people have complied. And it looks, at least from the data where, we didn't flatten the curve here, we kind of crushed it, luckily. So it's really, there's dozen or so cases, every day. And I'm not saying deaths, there's cases, detection of the virus across the country, maybe 20 or so on a bad day.
Yeah, it's quite a contrast to think about, though. I remember, Imran, me and you were at this gene and cell therapy conference in San Francisco when you were there at the beginning of March, and then I flew to the UK, and then when I came back from the UK into the US, I think was on March 15th. They basically gave me a piece of paper and said you need to self-isolate, and so I did that. My training's in microbiology, and I knew the importance of that, but I think most people were just given this piece of paper and they just tossed it in the garbage. So
were there fines in place, or?
I mean, we weren't really in a lockdown type situation yet at that point. So it's a bit different, but I guess, let's think about, now I guess Australia's kind of said, hey, this shutdown, whatever is,
cat.
Hey, Kitty. I think my cat joined us last week. So now we're in you guys are kind of going to this more transition phase, right? Because I guess Australia's kind of letting out. Yeah. So are you just raring to get back into lab because you learn these new techniques and play cricket again? And
yeah, no, it is. That's pretty much a situation. So yeah, there hasn't been formal huge changes with what we can and can't do, but the Prime Minister has said that we now need to flatten the unemployment curve is the next curve. So there definitely is going to be, I'd imagine, you kind of can't make a statement like that and not expect that people are going to start getting back. But you're right, I have to, without exaggeration, the time in Stanford, there was a week there was one of the most successful parts of my career so far, because we had these great breakthroughs over at Stanford working with the Porteous lab. And then also I got results back from a really nice CRISPR screen that I'd just done. So I had these two incredible results and really, really excited to get back and doing it. And so, yeah, raring to go. And so what I've pretty much done is just designed all my reagents. So a lot of that stuff is CRISPR-based reagents. And I think that's a good use of people's time. You can do these things carefully and slowly, whereas I think sometimes we do it pretty quickly, or carefully still, but quickly. It's nice actually to go through step by step and design everything. And also all the annoying jobs that you kind of put off. It's like, my least favorite thing to do is design, like you design guides and then you have to design the primers, so you can do the tide analysis. I always just skip over that step. But you can do that now, it's fine, and then there you go, we'll design the primers later. But you can do that. So yeah, I can't wait to get back to the lab, and we've ordered stuff and it's just coming, and yeah, we've got to wait. We have, some experiments going back. Some people can do some work at the Peter Mac, which is nice. And they've been able to do a few small experiments to get started. But yeah, hopefully we will be in a situation where we can really open the throttle up a little bit more, but we have to be cautious. We have to be very, very, very cautious.
So as you've been allowed back in the lab, I wanted to ask, what are some of the things as you're starting work that you didn't anticipate would happen? Surprises?
One thing, I'll answer this in like a personal thing. I was surprised at my own motivation levels. So I'm a very, very passionate researcher. I'm a very, very passionate scientist. And I, as you said, raring to go. But at the moment, I think I am emotionally not well a lot of the time. We're a little bit down. I did not expect to have to work against myself. And that's something I've not ever had to do. I don't always want to be there. And which is bizarre for me. It's bizarre. And it just sometimes when you turn on the news and you just see what is happening all around the world and the lockdown and the unemployment here and stuff like that, it can be a little bit depressing. So that's something, maybe just prepare yourself. You may be excited, you may be ready to go, but sometimes you have to fight against yourself, and I am fighting against myself quite a lot at the moment. Yeah.
So given that you guys are kind of ahead of the curve, compared to the US and Europe, actually, do you have any foresights about what life is going to be like in the lab when you get back, when everyone's back and working there? Do you see everyone wearing PPE all the time at first, you doing shift work?
Yeah, so I think one thing that will definitely probably have to be is shift work, and shift work is one of those simple ideas that you can really reduce your contact with other people. And you know, that can work, I guess, even with a Google Calendar. You just have a few people going in the morning and coming back in the afternoon. That's a good, I think, a reality of the future for going back. The other thing, of course, is just making sure you're not bumping into one another. And it's hard, you know, seeing your colleagues and not giving them a hug. That is weird. That is really, really weird to have not seen, you know, some of my colleagues for, you know, some of them 8 weeks and be, oh, hi, you know, like, you know, you want to give them a big squeeze, you know, and because I think we're all want a big of a hug. So doing science without having that physical contact is hard. And also we know that labs can be crammed sometimes as well. So that'll be a challenge, I think, going forward. Yeah, and keeping those distancing measures going the whole time.
Yeah. Great. So I think Aditya, I think we had some questions from the audience, right, that were submitted when people registered.
Yeah, let's get into that. And thanks, Imran. This is a side that I think it's nice to hear the human side of things, the emotional side of things. That is an impact this pandemic has had. And we addressed it a few times, and I think it's something that needs to be constantly spoken of because we are all human, and even though we're doing science, it's a key part of how we function. So that being said, we have some great questions from the audience. Shubra had a question. Will SARS-CoV-2 infected individuals mount protective long-term immune response?
Let the immunologist answer that one. Yeah.
Well, yeah, so it is a very, very good question. I think it's a question that's on everyone's minds at the moment. Now, I have to say I'm an oncoimmunologist. So one of these crazy things about my life is I split my life between cancer, immunology, and gene engineering. So I'm you know, my depth of knowledge of immunology isn't probably as great as some in the world, but there is some fantastic reviews if people do have these kind of questions. There was one released from Immunity yesterday or the day before. I think you'll be able to look it up on their website, and they do cover questions like this. And this is a really, really important question. I think the reality is there's no way of knowing whether we have a long-term immunity because we haven't had the virus around for a very long time. So we don't know what the response is going to be like. We know that there is obviously an antibody response and in patients that can detect the kind of antibodies that are consistent with a long-term memory. But the question is, how long is that going to stay around for? And so there have been studies done in rhesus macaques, and they can see that if they get one of these macaques infected with the virus, they are resistant for 28 days. So that's kind of promising. But how long is that going to go for? Obviously, we can learn some things from SARS-CoV-1, the original SARS epidemic and outbreak, I should say. And that they can look at the antibodies in those cases, but they do know that that wanes. So over 2 to 3 years, your antibody responses in those patients, people that did survive the original SARS, wanes over a 2 to 3 year period. And then on top of that, there's a good chunk of people, I think it's as much as 25%, don't have that memory response. So it's going to be something that we're going to have to keep in mind going forward, especially for those generating vaccines, that we may not have very long-term memory like we do for some other viruses. And people will be familiar with conventional vaccine genes. Sometimes you only need maybe 1 or 2 or 3 vaccines and you get a very long and persistent immune response. That may not necessarily be the case here, which is troubling, but you know, science will prevail, I'm sure.
So we have another few questions here from Mijal, that's from Takeda. What further actions should be considered due to the possibility of a bounce back in SARS-CoV-2 infections?
Yeah, I don't know if you have anything specific there, Kevin, you wanted to say. I think in the short term, for getting back to the lab, you have to do experiments that you can throw in the bin. You don't want to be setting up, you know, multi-thousands of dollars of experiments that, you know, in the case of an outbreak that you're just going to have to put in the bin. So I think, you know, a lot of my work is just doing some simple cell line work and not making some gene knockouts. That's actually really, really good thing I would recommend. Just doing a few simple experiments like that. If you have that as a part of your research goals, but because you can dump that in the bin and, you know, okay, you're going to lose time, but you're not going to lose thousands of dollars worth of reagents or animals if you're involved with animal studies. So, yeah.
Yeah, I guess that's a good point. Maybe the answer to that question is like don't get too comfortable too quickly, right? Because we don't know, and if that possibility does exist, like you said, maybe you got to bin a bunch of experiments, and but at the same time, maybe it's you just got to fight forward, right? I don't know. So yeah, I think it's something that like all the governments that are relaxing the rules now are really worried and concerned about that this possibility could happen. And are we opening things up too quickly? Right, it's a legit concern. Looks like we got another question here from Heather. What do you think we have learned from this pandemic? It's a really
I
broad
can
one.
give you a science answer or a politics answer, or
Let's do both.
Yeah, personally, I think science. I learned about from the science is that you need more than just science in life. That's kind of an interesting one. It doesn't make sense. Science doesn't make sense if not everything else is going, and we being separated from your family and stuff like that. How that doesn't work. I think from a political perspective and a more of a social aspect, I think it's interesting to see how vulnerable our economies are. And we have these, there's a fantastic article on the ABC here in Australia that covers this, but we have this kind of ready to go just-in-time economies where, you need to make something, so your product, your resources arrive on that day, and you generate it and you ship it off that day. And then as soon as you get any mild disruption to that, the whole system crumbles down. And I think, I hope that there is a bit of a look back and kind of go, can we make our economies and societies a little bit fairer and make a little bit more sense, and not just kind of be purely this, let's make profit is our only focus in every instance. And I think that's what this pandemic really has exposed is weaknesses in our society and in some political systems as well.
I guess the other thing is we've speaking about the science part of it. Maybe we've also learned the world in general as a whole, that how important the contribution of science is, right, to understanding viruses, doing research, infectious disease research, understanding epidemiology, tracing patterns, things like this, and vaccine development, right? And maybe we're underfunding all of those things. I think as scientists, it's always very easy for us to sit here and say, yeah, we need more funding. But like when stuff like this happens and the whole human race is affected, I think everyone else can see, hey, maybe it's actually more important that we pay attention to this and we educate ourselves about this rather than following, the Twitter feed of the most famous socialite or something. So yeah. I think hopefully that's something that's going to come out of this, that the whole world has kind of learned how important it is for us to be better at science.
And Kevin, we were prepared. Like we had warnings about this. We had multiple mini outbreaks of various different diseases, and over the last, in my lifetime, I can just think of quite a few. Just SARS and MERS and the few different flu strains that we've had. Obama was talking about this a long time ago about the necessity to prepare for this, and we weren't. We, like most governments around the world, were found wanting. So yeah, it certainly has taught us that we need to take these things a little bit more seriously. That's for sure. Yeah.
Great. Another question from Ronald Northeastern University. How to select sgRNA targeting sequences in my genes of interest?
All right, back to the basic CRISPR science. Imran, how do you do it?
How do I do it? I use the Synthego tool a lot of the time. Excuse me. I use the Synthego tool a lot of the time because there's a function there that I really like, in that it looks at different exons. And I'm not trying to sell you a tool, but that's the reason I use it, is that you can use it, can make sure that the exon you're hitting is expressed in multiple transcripts. I do have to say I do check them. Because a lot of the time in my strategies, I'll hit multiple exons at the same time, and I'll go back and do some basic things on benchling as well, just to target other exons. So put that on top of that as well. Oh, there we go. We have a link of the design tool in the chat. For HDR guides, it's like for inducing fragments, it's a little bit more complicated, and that kind of, you have to be shown, I think, how to do that, because there's quite a few things for you to consider as well about getting rid of PAMs and making sure you select the safest guide as well if you're going to do clinical application as well.
So we actually have a question coming from the audience from Daylin. I'm currently an undergraduate biology major on a pre-med track, and I'm very interested in CRISPR. Do you have any recommendations as to how I can get started involved in this field of research?
I guess like utilizing CRISPR in your research is I mean, there's a lot of resources out there now. I don't want to plug it too much, but just Synthego, we have a ton of resources on our website now and learnings you can get. There's also, a lot of other good resources out there. I know AdGene has a lot of good stuff as well. So, and then, I think really it's a lot easier to educate yourself now about the CRISPR technology, how it works and what doesn't work by checking out those resources. I'd also recommend, picking up some primary literature. Actually, we have a CRISPR journal now as well, so that's great. So it's not just, random articles in different journals that you can read about CRISPR, and there's an actual dedicated CRISPR journal. So that's a good way to get into the field as well, I think, just to kind of see what other people are doing.
Great. So I think last question here, how can genome engineering help us fight COVID-19?
Yeah, it's a good one.
Yep.
No, you go for it, Kevin.
I mean, there's definitely a couple ways. So, on this show, the CRISPR Office Hours, for a couple of weeks ago, we had both Ted from Mammoth Biosciences, and then we also had Jonathan Omar from MIT talk about how they've been using the CRISPR technology to build diagnostics to detect SARS-CoV-2 RNA. And so actually, I think just this morning, the Sherlock test got EUA, so FDA approval, and that's a Cas13 based assay, right? So it relies on the Sherlock technology. So that's one way we can use CRISPR using some of these Cas nucleases to detect it in the environment, detect the RNA in the environment from like a swab or something like that, or saliva. And then I know they have a new test that they've just developed as well at MIT that's on their Stop COVID webpage. So that's worth checking out. We actually also have a webpage too. You can look at. I think if you click on, we probably have a link to it here. So if you click to the next slide, you can probably see the link to it. Right, but if not, it's synthego.com/coronavirus. And also, we've been collaborating ourselves with a group that has looked at interactions between viral proteins and human proteins. And essentially, if you knock out some of these genes, what you can do is you can actually see which ones are related to the viral proteins themselves and which ones are actually interacting. And so being able to use that kind of a resource like CRISPR knockouts, which is a very easy thing to do with CRISPR, relatively easy, you can basically look for drug repurposing and validate your drug targets, or you can also, test to see which genes are basically essential for the viral infection. So if you could do a viral infection study. So those are just two ways that you could use CRISPR, either through a diagnostic approach or actually genome engineering cells to either validate if these genes are associated with the virus in any way, or if they are good drug targets.
Yeah, you stole my answer, Kevin. The second part was going to be my answer. So, no, I think definitely from a research perspective, you can see that you could very easily modify, cells that have been infected by a virus, and just seeing what molecules are important for infection and potentially resistance if you get lucky. So yeah, I think from a research application, there's a huge range of different things that can be done there.
Great.
Looks like your cat wants to contribute to the answer again.
He does, yeah, he wants to contribute to every moment of my life. But anyway, that's fine.
All right, so maybe just we're starting to wrap up now, and maybe just some quick takeaway thoughts, Imran, from as we go into this new normal. What do you think your new normal looks like?
My new normal, it's going to be interesting. I think it's my new normal at the very short term. New normal is going to be making sure that everyone in my team can stay motivated. Everyone who I work with, I guess even above and below me, are ready to go back. And can, from an experimental perspective, what experiments we can focus on to get everyone back in the lab and going, but also, from a mental perspective, how is everyone going to get in the right mindset to go? It's very weird to be in an apartment for a long time by yourself, to then go back into a bunch of lots of people together back in the research department. It's got to be a shock, and just making sure everyone's looking after themselves. Yeah.
Yeah, that's definitely key. Looking after yourself and the emotional side of things and self-motivation, definitely key. So as we're beginning to wrap up office hours this week, wanted to ask Kevin, who's going to be on office hours for us next week, May 14th at 9 a.m.
Yeah, so if you want to move to the next slide, we're going to be joined next week by Kiana Aron, who is an assistant professor at the Keck Institute in Los Angeles. And she's also the co-founder and CSO of Cardia Bio. So, if you haven't heard the story yet, she developed the CRISPR chip technology using, which is pretty incredible stuff, merging transistors with CRISPR technology. And she's going to be talking about basically the next generation of CRISPR-powered transistors that maybe we can use for detection of respiratory pathogens and other pathogens. So should be a pretty interesting and exciting talk.
Great. Looking forward to it as always. And so for the folks who are online, to get your Synthego Keep Calm and CRISPR On shirt, just go to synthego.com/calm and you can fill in the form and then claim your shirt. So just want to say, as always, thank you today to our special guest, Imran House from Down Under at, I guess now, 2:50 a.m. We really appreciate it. Hopefully you can shut eye after. And as always, it's a pleasure to host with you, Kevin.
All right. Yeah. Thanks, Aditya. Imran, thank you very much for joining us today. And thanks for all your insights. It was really interesting to hear your experiences, what happened to you, and how you're ready to get back to work. And, hopefully we wish you all the best of luck and that you stay safe. Everything in Australia works out great for you.
Thank you so much. And good luck to everyone.
Thanks, everyone online. Look forward to seeing you guys next week. Bye.